This video will tour two specimens of the thymus. First, the one on the left here. And this, the look of the thymus is blue. Dark blue, light blue. The cortex are rims of blue, very dense staining blue, and the medulla is lighter, with of course the specialized structures in the medulla, the Hassel's corpuscles. So if we zoom in on, let's say, the cortex here, each of the thymic lobules separated by connective tissue trabecula are composed of a dense area of lymphocytes in the cortex and a lighter area, less density of lymphocytes in the medulla. These are the Hassel's corpuscles here, and all of these cells in the medulla are the native T cells that are have been processed. They're either CD4 or CD8 T cells that have been processed in the cortex. And your lecture pretty well spells out that process. I think this annotation shows a section of the cortex over here, and of course I just illustrated another section of the cortex. All this area in here is the medulla, and then I think that we picked a fairly typical Hassel's corpuscle, which are actually collections of dead epithelial reticular cells. You may recall from the lecture that the entire framework of the thymus are epithelial reticular cells connected by desmosomes that form a framework much like reticular fibers do in the liver and the spleen and the lymph node. And so this forms a framework, and it also they wrap around the blood vessels to provide a barrier, particularly in the cortex, where we may have a better example of that barrier in the other specimen. But so I was just about ready to say that the typical, very diagnostic feature of the thymus, if you can look in the medulla and find a Hassel's corpuscle, you know it's the thymus. But you also, in the cortex, you don't find any nodules. You just find the cortex area, which is very dense lymphocytes, and the lighter density of lymphocytes in the medulla. So let's now shift our attention over to the other thymus specimen. And this specimen is probably a little cleaner and maybe a younger individual, where you can see the outline of the cortex here and the medulla, very nice. You can see the Hassel's corpuscles here. There's one over here. I think I see some more over in here, and then of course these are blood vessels here. And in the cortex, when you find a blood vessel, it's surrounded by epithelial reticular cells, one layer, which are phagocytic cells, and then an endothelium. And you have a very distinct base membrane, and this blood vessel is isolated from these cells being recognized, the white cell membrane makeup, as well as erythrocytes. So it does not; these cells do not get programmed to destroy normal body cells. And of course, as you learn in lecture, there is a whole sequence here where you have macrophages here that actually take out and recognize T cells that are going to recognize normal body cells. So there's quite a process that goes on in the cortex here, and then finally you get the native T cells, the CD4 and CD8 cells here in the medulla that get out in the bloodstream. So be sure to study these specimens and relate what you learned in the lecture and the lab. Okay, let's end this one.